Hello everyone, my name is Crystal Santos and I'm excited to be doing my creature feature project on the Limulus Polyphemus, also known as the Atlantic horseshoe crab. Despite their name, horseshoe crabs are more closely related to scorpions and spiders than actual crabs. They have been roaming the earth for over 450 million years and have survived 5 mass extinctions, making them older than dinosaurs. They are living fossils and have remained nearly unchanged since they appeared on Earth, due to some exceptionally effective adaptations and genes that code for remarkable molecules that have allowed the horseshoe crab to survive, just as it is, for so long. Despite their strange appearance, horseshoe crabs are harmless. They do not have any stingers, fangs, or teeth, and they are not venomous. If you ever see one washed up on the shore, never pick it up by its tail. Their tail is what they use to right themselves when they are flipped upside down. If you break its tail, essentially it will die, as it will expose them to predators. This is an image of my daughter Jalen last summer learning about these wonderful critters. The Atlantic horseshoe crab lives along the coast of the United States in the Atlantic Ocean. They can also be found in the east and Gulf Coast of the United States, as well as Mexico. Horseshoe crabs utilize different habitats depending on their developmental stage. The eggs are laid on the coastal beaches in late spring and summer. They're usually found in the sand or mud flats, estuaries, and mangrove swamps. Horseshoe crabs can survive a wide range of environmental conditions, including variations in salinity. After hatching, the juvenile horseshoe crabs can be found offshore on the sandy ocean floor of tidal flats. Adult horseshoe crabs feed deeper in the ocean until they return to the beach to spawn. In the winter, they can be found at depths of about 30.5 meters, which is 100 feet. The two greatest threats to horseshoe crabs are habitat loss with coastal development and shoreline erosion, as well as overharvesting. The horseshoe crab has a horseshoe-shaped, hinged, hard shell body that is divided into 3 parts: the carapace, or the head region, middle abdominal region, and the testline tail. One of the reasons natural selection has preserved them is their hard shell, called the carapace. It's so strong that only sharks and turtles can penetrate it. Guiding them through the ocean are 9 eyes, 2 compound eyes that act much like our own eyes, 5 secondary simple eyes on top of their shell, which can detect UV, and 2 ventral eyes located on their underside, perhaps to help them with orientation. Along with their complex circulatory system, 5 pairs of gills, and 12 bristle legs, evolution created them to be creatures extremely well adapted to their particular environmental niche. The color of the crabs range from light brown to black. You can identify if a horseshoe crab is a male or female by looking at its first set of appendages. The female have pinchers and the males have little punching bag, glove-looking appendages. The females are also larger. In this image you can see that it's a male because of its first set of appendages. The horseshoe crabs go through a molting process. The molting process requires shedding small exoskeleton in exchange for larger shells. They go through 16 or 17 molts during their development. At around 10 years of age, a horseshoe crab reaches adulthood and they are ready to start breeding. A horseshoe crab can live for more than 20 years. Beyond evolution creating physical traits that make them so well adapted, much of their survival is due to something we can't see: their simple but incredible immune system. It has protected the species from bacterial infections for eons. In 1956, a medical researcher named Fred Bang at Marine Biological Laboratory in Woods Hole, Massachusetts, discovered that blood cells from horseshoe crabs vigorously clot in the presence of bacterial endotoxins. The blood cells of a horseshoe crab consist of the substance called Limulus amoebocyte lysate. Fast forward to 1977, and the United States Food and Drug Administration has approved LAL to test drugs and vaccines because any amount of endotoxins in a drug or vaccine can be fatal to humans. The LAL blood test is the worldwide standard for screening for bacterial contamination. Today, every drug certified by the FDA must be tested using LAL. A key characteristic of this species is their blue blood, which is due to having copper in the oxygen transporting proteins called hemocyanin. Every year when the horseshoe crab arrives on the beaches of the Atlantic coast to lay their eggs, hundreds of thousands of them are rounded up and brought into a lab to be harvested. Not to be killed, but for their blood to be carefully extracted. Their blood is so valuable that is the basis for a multi-million-dollar pharmaceutical industry, and a single liter of it goes for around $16,000, making it one of the most valuable liquids on Earth. Endotoxins remain stable at high temperatures that even an autoclave will not break it down. Even if the bacteria is dead, the endotoxin will remain. When creating injectables, healthcare products like vaccines, medical implants, and IVs, it is imperative that they are free of any invading microbes. It's pretty clear that horseshoe crabs are vital to human health, medical research, and safe vaccinations. I would argue that if it wasn't for horseshoe crabs, we wouldn't have the ability to combat the COVID-19 with the safe vaccinations. Our reliance on these animals puts immense pressure on a fragile ecosystem, and so far scientists have struggled to recreate this compound and its effects in a lab. We need to protect this prehistoric species as it plays a critical role in the coastal ecosystems as well as providing a huge benefit to humans. Thank you for listening to my presentation.